Welcome to the Chapter 2 podcast. The nervous system reaches into every part of the body and coordinates every aspect of bodily functions, from seemingly automatic processes like heart rate to the most sophisticated behavioral responses and complex emotions. The immense amount of information processing that the nervous system performs requires an immensely complicated computer. More than 80 billion neurons with widely varying shapes and sizes, reflecting the functions they perform. Nevertheless, neurons can be categorized according to general layout, size, and function. Typical neurons share three main structural elements: dendrites, which are the main receptive surfaces of the cell, a cell body containing the cell's DNA within the nucleus, and an axon, which carries nerve impulses away from the cell body. From a functional perspective, neurons share four basic elements. 1. An input zone, consisting of dendrites and cell body. 2. An integration zone, the axon hillock. And 3. A conduction zone, the axon. And finally, 4. An output zone, the axon terminals. Glial cells also serve important functions in the nervous system. They myelinate axons, exchange nutrients and other substances with neurons, remove cellular debris. More recent data have revealed that glial cells also contribute to information processing. Information is transmitted from a neuron to other neurons, or to muscles or glands, across specialized contacts called synapses. In most neurons, the axon terminals contain specialized boutons that release molecules of neurotransmitter in response to the arrival of an impulse along the axon. The neurotransmitter crosses a synaptic cleft and alters the electrical properties of the postsynaptic cell via receptor proteins that recognize and react to a specific neurotransmitter. The brain can be visualized in many ways. Static images of cells within the brain are achieved using different stains that bind to different types of molecules. Labeled antibodies can be used to bind to and visualize cells producing specific proteins. In situ hybridization allows for the visualization of cells producing specific messenger RNAs. White matter, axonal or dendritic tracts, can also be visualized using specific stains or labeled molecules that are injected into one region of the brain and transported by neurons. Neurons can be classified according to their size and shape or by their function. Neurons that bring information into the spinal cord and brain are called sensory neurons. And motoneurons, or motor neurons, bring information to muscles. Interneurons make up most of the brain and spinal cord and are important for integrating information from other neurons. The nervous system consists of the central nervous system, the brain and spinal cord, and the peripheral nervous system, the sympathetic, parasympathetic, and enteric divisions of the autonomic nervous system, the spinal nerves, and the cranial nerves. A simplified view is that the sympathetic nervous system prepares body systems for action, while the parasympathetic division has opposite effects tending towards relaxation. The enteric system regulates functioning of the gut. The spinal nerves bring information to and from the spinal cord, and the cranial nerves carry information to and from the head and neck. In order to discuss the relative position of brain structures, one must understand the terminology used for orientation. In general, positions are identified as paired opposites. Top and bottom are referred to as dorsal and ventral respectively. Medial and lateral refer to closer to the midline or to the side respectively. Anterior or rostral refers to the front or toward the nose, and posterior or caudal refers to the back or to the tail. These terms are easiest to understand in a body plan for animals that walk on all fours. When thinking about these terms in humans, think about a person as an animal that has stood up. For example, dorsal is still towards the back, and ventral is still toward the belly. The most prominent features of the brain are the two cerebral hemispheres, the surfaces of which are heavily convoluted with gyri, humps, and sulci, cracks. Each hemisphere contains the four major lobes, frontal, parietal, temporal, and occipital, made up of gray matter, cortex, overlying white matter. Underneath the cortex, integrated systems, such as the basal ganglion limbic system, can be found in the forebrain. The brain first develops as three distinct regions. 1. The forebrain, or prosencephalon. 2. The midbrain, or mesencephalon. And 3. The hindbrain, or rhombencephalon. The forebrain differentiates into two regions called the telencephalon. Cortex and basal ganglia are prominent, and diencephalon. Thalamus and hypothalamus are prominent. The midbrain remains as one region. Superior and inferior colliculi are prominent. And the hindbrain differentiates into the metencephalon, the cerebellum and pons are prominent, and the myelencephalon, the medulla, is prominent. The brainstem is considered the midbrain and hindbrain together without the cerebellum. Think of the brain as a flower, with the forebrain being the flower and the midbrain, pons, and medulla as the stalk. The cells of the neocortex are arranged in 6 distinct layers, with different layers performing different functions. Some other telencephalic structures are made up of allocortex, which only have 3 layers, or an unlayered organization. Cytoarchitecture varies within the cortex and seems to correspond reasonably well with the functional organization of the brain. Clusters of cortical neurons are organized as functional units within cortical columns. Thus, information is segregated, horizontal to the surface in layers, and perpendicular to the surface in columns. The brain and spinal cord are protected by the 3 layers of meninges, the dura mater, the arachnoid, and the pia mater, and are suspended in cerebrospinal fluid, which is produced in and circulates throughout the internal ventricular system of the brain. The brain is richly vascular, supplied by the anterior, middle, and posterior cerebral arteries, plus numerous arterioles and a backup route called the circle of Willis. An alternation or disruption in the blood flow in one of these arteries, for example due to a stroke, causes a lesion in the brain region supplied by the affected artery with concomitant deficits in cerebral function. There have been significant improvements in techniques that allow functional and structural study of the living brain, such as CT, MRI, DTI, functional MRI, PET, MEG, and optical imaging, along with transcranial magnetic stimulation. Using dyadic functional MRI, researchers can image brains of two people simultaneously as they interact side by side. And this concludes our podcast.